Hello everyone, Priyam this side from the Department of Bioinformatics, MMBBHU. We have been given the task to choose and demonstrate tool related to systems biology. So here I'm gonna do just that. The tool that I have selected are GraphWeb and APIT. GraphWeb is a public web server for graph-based analysis of biological networks that analyzes directed and undirected, weighted and unweighted network of genes, proteins, and microarray for many eukaryotic genomes. So GraphWeb is designed only for eukaryotic genomes. It can also integrate multiple diverse data sets into global networks. It can also filter nodes and edges based on dataset support, age, weight, and node annotation. It can also detect gene modules from networks using algorithm. We have so many algorithms in GraphWeb that we are going to see in the demonstration part. And also gene modules are very important in network biology because they perform a biological function in an autonomous manner. They are basically a subnetwork of more closely related nodes, and they may act as functional unit, biomarkers, and target for drugs. GraphWeb can also interpret discovered modules using gene ontology pathways and cis-regulatory motifs. The next tool that I'm going to be talking about is APIT, which stands for Agile Protein Interactomes Data Server, and it is a unified protein-protein interactions data server which uses data from known databases such as Bind, BioGrid, DIP, Mint, etc. It provides a comprehensive and curated collection of protein interactomes for more than 1100 organisms, including 30 species with more than 500 interactions. And the data is derived from the integration of experimentally detected protein to protein physical interactions. Here are some journals mentioning APIT and GraphWeb. Now let's talk about why I have chosen these tools. So the first reason being both the tools selected by me are web server based and do not need special skills and system requirements to run. Both the tools are easily accessible by just a simple click of buttons. APIT provides a unified platform to view unified results from several renowned databases like Mint, DIP, HPRD, saving so much time. GraphWeb is designed to analyze individual or multiple merged networks and can also be used to search for conserved features pointing towards evolution. And GraphWeb can also compare results of high throughput dataset. Now let's move on to the demonstration part. So now let's go to Google and type GraphWeb here. We have to click on the first link. Upon clicking the first link, we will be greeted with the interface of GraphWeb. As we have already discussed, GraphWeb is a public web server for graph-based analysis of biological networks. So we will be greeted with three columns. The first one is for defining the network dataset. The second one allows us to choose network algorithm from a variety of algorithms like connected components, MCL clustering, betweenness, centrality clustering, etc. And the third one is for modifying the network settings. It allows us to remove unknown and unambiguous genes, create global networks, hide modules with less than however nodes that we want, sort modules by functional score, etc. So it also has option to put data from direct input. We can also upload a file in our computer or file from the server that it is mentioning. But let's move to the advanced input option here. From here, I will choose example input because I just have to show the data. Then I'll add input. We'll wait for it to load. Then we will click on finish. Now let's choose an algorithm. So let's do the first one, connected components first. Let's click submit. Let us wait for it to load and then we will be able to see the results. I think the results are here. Let's scroll back. Here you can see the network information. It tells us that the network type is undirected. It is having 3,274 nodes, 5,562 edges, with edge density of 0.1% and average node degree of 3.4. It also tells us that the clustering coefficient is 0.027. Also, the module information shows that the organism is Homo sapiens. An algorithm that we have selected was connected components. It also shows the nodes name in conversion table. Upon clicking it, we can see all the nodes IDs and input IDs. We can just copy paste all the data from in here. We also got information about the modules found in this network, which are 110, and out of them 25 are large and significant. So it is showing only that. If you are interested in like studying for a particular node, you can search for that node in here. We are also having scores, and we have option to see the nodes and edges in that particular network from here. We can just click on here and then we will get the data for all the nodes, which are 3032. We'll get the information for 3032 nodes from this file, and the edges from this file, which will open 5428 edges. Yeah. We can also view the data in like visual form by clicking here. Here it doesn't give the option to see the visual, but here I have the option to see the visual. Let's click on this compact thing. Here we can see the network, but it is not labeled because we have chosen the compact version of it. Let's click on labeled one now. Yeah, so we have the same network, but it is now labeled with genes. We can like show it in different versions like SVG. We can also get coordinates for it, and we can also choose different layouts such as dot, circle. You can play around with this web server. Also, you can export all the data from in here. One more thing, you can get all this information from G Profiler, which is used for functional enrichment analysis. So let me just do that. Let us copy this data and let's execute G Profiler. Let me copy paste this data here and then we will run our query. So it doesn't give any information. We'll try again with another example. Let's go back here and this time let's click on a different algorithm. Let's do hub-based modules or maybe maximal clicks. Let's do hub-based module first. Click on submit. Scroll back. So yeah, data for hub-based modules are here, and it is showing that the modules find are 3274. That's why it is taking so much time to load. Let's wait for a minute. Meanwhile, let's look at another algorithms which are present. So we have strongly connected components, hub-based modules that we have selected just now. We have whole graph, we have bi-connected components, we have maximal clicks, we have MCL clustering, we have between the centrality clustering. We have so many options to choose from. Also, we have network settings where we can assign more weight to smaller networks. We can also create global networks. We can also remove edges. We can remove genes with unknown and unambiguous functions. We can do so many things from this web server. It is still not loaded. Meanwhile, let's look at the APID. Let me just Google search it. Agile protein interaction database. And then I'm gonna just click here on the second link. Upon clicking, I will be greeted with the interface of APID. Yeah. I think the results for graph web are here. Let's click on it. So yeah, there is so much modules that it is not able to load. So let us try it with different algorithm. Let's do betweenness centrality. Let's do sample size of 50 because we have so much data, and let's remove 10 edges at once. Now let's click on submit. Meanwhile, let's explore APID. So APID is basically an interactome data server, and we can like upload data that we have about interactomes. We can search for a single protein. We can also search for a list of proteins. We can search for publication, etc. So let's do one protein first. We can also give this name that it is given as example. Let's put it. Let's put KRAS here. Then we search it. See, we have found three proteins for KRAS. The first one is in human, the second one in mouse, and the third one in rat. So in human, we have 891 interactions, and in mouse, we have 15, and in rat, we have 10. We can also display these results in the form of interactions, in the form of procuration events, and as network. But network will be too hectic because it is of 891 interactions, and it is unable to show interactions above 200. So let's make a network for mouse one. We have to click on this circular thing. See, we have got the data as a network here. An interesting thing is it is a unified database, so you can get all the information about genes from proteins database. You can get information about functional environment from Go. You can get information about structural environment from Interpro. You can get information about structural environment again from PFAM, and you can get information about pathways from Reactome. So you can get all the data without visiting these databases one by one. We can just get it all together from this one platform. Also, we can choose different network layouts such as grid, circle, concentric, whichever suits our needs. We can also like filter the networks according to experimental evidences, which will show as blue color in here. We can also expand the network. We can export it in the form of JPG, PNG, or TXT. Also, we can copy paste this data. One more thing, if I click on a certain node, I'll get the information about that particular node, like its uniprot ID, its name, and its description here. It will also show the number of interactions present in it. We can also load the like related interactions from here. Here we will be able to see all the interactions which are showing in mouse because we have done only mouse now. So yeah, APID is very useful because in one data server, we can get information about proteins, pathways, etc. without visiting any of these databases independently. Let's see if graphic results are out or not. So yeah, we have web results. So it has found 419 modules by using the algorithm betweenness centrality clustering and Homo sapiens. And here we have the data. Here I am having all the data, whichever I want to see. Let's see this compact version of network. Let's see its labeled one. So now you can like do profile annotations, you can visualize it, you can get the data about nodes, edges, etc. all in one using this graph web server. Let me do this G profiler for you. I'm just going to copy this data. Then I'm going to execute the G profiler. Let me put this data here and let me run a query once more. Still, it is not showing any results. Let me try it with another one. Let me do this time. Let me execute JProfiler once again. We put this and that's ready. Still no results it is showing. I don't know why. Maybe it is not having any data to show. Let us try another one. Let us try MCL clustering now. Let's submit our data. So this time we have got data for MCL clustering and it has found 658 nodes this time. Let us spot modules by functional score so that we are able to see more like important modules at first. See, we have got the results for MCL clustering and density is varying between all the modules. We can get information about nodes and edges as I have previously showed. Let's execute GProfiler this time. Let's copy this data. Let's hope it will run this time. Yeah. Oh, I have got the data this time. So it is showing the functional enrichment analysis for this particular gene that I copy and pasted from there. So yeah, without visiting any other like data server or any other thing, we can get the functional enrichment analysis directly from Graphic. And it is showing results from cake, reactome, etc. from like very robust databases in bioinformatics. Uh, that will be all. Thank you. Thank you for listening up to this time.